Hi, good afternoon, I guess, technically. Thank you all for having me here today. I do have to preface everything I say with the disclaimer that appears in the lower left-hand corner there that I'm here speaking as a scientist from the EPA Office of Research and Development. I actually don't know anything about policy at the EPA, and anything I tell you is what I told you. It's not what the EPA told you. So, disclaimer. Blame me, not the EPA. So, like I just said, I'm from the Office of Research and Development. I get to have a lot of fun today here because I'm presenting work that's been done by dozens of collaborators at this point. I do want to just say the Office of Research and Development is quite large. Our single largest facility is in Research Triangle Park, North Carolina. We published 620-something papers in 2017. We've officially only 562 so far in 2018, but I think that's because our database is a little slow, not that we've gone down. And then we have 13 facilities throughout the United States, and I work with federal researchers, contractors, uniformed members of the Public Health Service, contract researchers. I have a graduate student at University of North Carolina. I've mentored students at North Carolina State, so it's a diverse group down there. So, title of the talk, refers to moving beyond the lamppost, and this is really right back to where Dr. Hartman was talking about right at the beginning today of what else is out there. And, the old expression, where do you look for your keys? Well, you look under the lamppost because there's light. Well, did you lose them there? No, I lost them over here, but there's no light, so I can't look for them. What EPA and our group in particular is interested in is not ordering the deuterated standard when we get the call because we know everybody's going to be interested in 1,4-dioxane. It's anticipating what that chemical might be before we get the call, before USA Today tells us to look for it. And so to do that, we really need to get beyond the lamppost of what we're looking at. And so in the United States, there's basically a tapestry. There is no single chemical law in the United States. It's a tapestry of laws. That's actually former Justice Breyer, who was writing on how we regulate chemicals. And depending on what the use of the chemical is, there's very different testing requirements. So a pharmaceutical, a drug active, has very stringent requirements. It has to be proved to be safe, effective, also be human testing. Food additives have significant testing requirements. Pesticides, which are designed to actually kill something, have very significant additives. However, there are many chemicals that are not, it turns out, food additives or pesticides or pharmaceuticals. And so in a study back, Park et al. Is the first author, is done by Dean Jones' lab at Emory University back in 2012. They looked at pooled blood, and they saw markers of over 3,000 chemicals. Now, some of those chemicals in the blood are blood, probably most of them. Cholesterol is the most abundant thing. Maybe that's a separate issue of what do you want to do about that. That's not technically the EPA's problem. But what we want to know is how many of those chemicals are exogenous? What do we know about how they're used? What can we do about them? So there was an issue of New Scientist magazine back in 2014. Everyone I know can argue all day about what the actual number here should be. They claim we've made 150,000 new chemicals. We touch them, we wear them, we eat them. Which one should we worry about? The correct number is certainly in the tens of thousands. I don't know what the exact number is, but the idea is they show a bright red shirt, some lipstick, and an apple with a bite out of it, and the idea is chemicals that we've manufactured, chemicals that we want are in all those products. What do we know about them? So most of the chemicals that are not food additives, not pharmaceuticals, not pesticides are regulated under something known as the Toxic Substances Control Act, or TSCA. And when TSCA went onto the market, or went into law in 1977, there were thousands of chemicals that were effectively grandfathered in as well. We're doing okay right now in 1977, so good. What turns out, there's never necessarily been toxicity testing, exposure assessment, or pharmacokinetic assessment of those chemicals. Now, again, we're standing here 40 years later, we're doing okay. So it's not like the end of the world, but basically, the U.S. Government Accountability Office describes that there are tens of thousands in commerce, and we get requests to add about 600 more under TSCA every year. And these are not getting animal testing or human clinical trials. What we do have are high-throughput screening tools that have largely been driven by drug discovery, and they're very biased toward drug discovery. They're really good at telling you whether or not something's a drug. What they can tell you about bisphenol A is, has been debated a little bit over the past. But two of the major programs are Tox21, which is a consortium between the US FDA, the EPA, and multiple institutes of the National Institutes of Health, including the National Toxicology Program. At EPA in particular, the ToxCast project has cast a wider net for looking more in depth at bioactivity across fewer chemicals. So only 2,000 something are published right now. We've got another 2,000 that we're working to release. All of these technologies are based on basic ideas of you can have a silicone plate, and this actual plate would be about the size of my hand, a little bit smaller, have hundreds of wells in it, and you can design an experiment with a single readout. So for instance, you might use some sort of green fluorescent protein, and so if you have your positive control in these wells and you see a nice bright green signal, you put your test chemicals and titration down here, and so you're increasing the amount of chemical. And so you see you got 1, 2, 3, 4, 5 chemicals here that seem to give that green fluorescent protein signal that our positive control gave. And so you're able to just use a robot, pipette in your chemical and dose in concentration response here, and take a single picture and get a ton of data out of that. Now, in vitro and vivo extrapolation, all the interpretation of this is very complicated. My center has been working on this for 10 years now. It's not easy. In fact, if this chemical here is a fluorescent chemical and we just happen to put it on the plate, that's a real problem, probably doesn't indicate anything. But all of that data has been made publicly available through a series of publications and then also a public dashboard where you can basically search, drill down to any one of these assays, and we've literally conducted thousands of assays across almost 10,000 chemicals at this point now. So taken together, we call these new approach methodologies, and the TSCA law, the Toxic Substances Control Act, was updated in 2016 to allow EPA to start to explore evaluated new approach methodologies to assess toxicity. And so what we, for instance, one of the early accomplishments we had, and this is shown by this figure, and I guess I should be don't not be so biased with my laser pointer, so both on the left and the right here. We were able to replace the rat uterotrophic assay. So that's an idea where, or that's an assay where you actually look for estrogenicity of a chemical. It was conducted in vivo. We were able to replace that with a suite. There's no single one in vitro assay that miraculously replaces an animal study yet, but a suite of assays was able to very successfully replace the rat uterotrophic assay, and so that's actually EPA policy now that one is replaced, and we're working on identifying additional chemicals for testing using things like the rat uterotrophic assay. Now that's one assay. Anybody here who's familiar with toxicology is aware that there's a lot more than one animal assay, but it's a very good first step. So I'm here to focus on, so that was ToxCast, and the EPA lingo, that's the Toxicity Forecaster Project. And just like with weather forecasting, if they say it's a sunny day, you sometimes still bring an umbrella if it's important. That's the sense of toxicity forecasting. It is a forecast. The ExpoCast project is the exposure forecasting project. And so with ToxCast and work in high-throughput Toxicokinetics, again, from the pharma industry and driven by Dr. Barbara Wetmore at EPA, you can convert concentrations from an in vitro assay. What concentration caused that assay to glow green to milligrams per kilogram per day that a human might get exposed to. And so here we have a couple dozen chemicals, about 50 monitored by the Centers for Disease Control and Prevention. And we have in vitro estimates of what dose you would need at a steady rate to cause the bioactivity that's been observed by ToxCast. And so we have a lot of chemicals up here. We're talking in mg per kg per day. And I'm sorry, I've covered up most things, but this is about at 1 mg per kg per day. It's actually quite a lot of the chemical. And then you see some of these are pretty uncertain from ToxCast of, well, really, what concentration do I need? I don't know. I only know plus or minus 100. That seems very uncertain. However, when you look at the exposure numbers can be really bad. These are exposures inferred from the NHANES study, National Health and Nutrition Examination Survey. These numbers can be really uncertain too. I don't really know how much of this one chemical here I'm getting exposed to. However, if we've done a good job of characterizing the uncertainty, I can be uncertain in the toxicity, I can be uncertain in the exposure, and then I can look over here and I can kind of call this the graveyard of graduate students. If my advisor says we're working on this chemical over here, well, I don't know anything about it, but I do know that it seems like bioactivity and exposure are separated by 100 million-fold. So that means that the likelihood that my correction and my research is going to refine things to the point that we're actually concerned about the impact on the average population is smaller. On the other hand, there's two chemicals here where, again, ToxCast needs interpretation. These exposure numbers are not ideal, but there's some plausibility of an overlap for these chemicals at the far end here. Those are chemicals where, if I was a graduate student, I'd feel much better about putting my time and effort into studying them. And that's the sort of prioritization we're working on. So this is just another schematic of outlining what we're talking about. We want hazard information, we need toxicokinetic information, and we need exposure. And don't just believe me, this is actually out of the National Research Council back in 1983, established this way of looking at things. And so if we have high-throughput screening for toxicokinetics and hazard, and that's been a development of the last 10 years that a ton of effort has gone into. And so whether or not you believe those tools, my job is to say, okay, somebody else has figured out how to do this. What numbers do we get for exposure? We do not have high-throughput exposure tools, or we did not when we started the ExpoCast project. Most chemicals lack public exposure-related data. So we have tens of thousands of chemicals that are on the U.S. high production volume list. That means we produce them or import them at more than 25,000 pounds in a given year. So we know that. Then this is on a log scale. We very quickly drop off. About 10% of those, we actually know what they're used for. And when we get down to biomonitoring data of what's actually in people, we have hundreds of chemicals. Now that's monumental effort. You've heard all the work today on how hard it is to develop a method to quantitate a chemical, and that's often in an extract or something. If you had human blood and you have to subtract off the Big Mac that I ate last night, it gets complicated. But the fact is we have a major mismatch of orders of magnitude of what chemicals are in commerce and what chemicals do we actually know are in people. And this is, yeah, again, not to knock the CDC. I think it's the NHANES survey is amazing. Basically, it's a rolling study that's been conducted since the late 90s, every 2 years. They look at roughly 10,000 Americans. They currently divide those 10,000 people into 3 groups and look at the chemicals in the blood of each of those 3 groups, because there's only so much blood sample to go around. But you get things like what phthalates are in the blood and urine of these individuals, what organophosphate pesticides, what pyrethroids are in there. They have covered hundreds of chemicals and have characterized the average American with respect to what's in there. However, if we go back to those studies by that group of Emory, they're seeing greater than 3,000 chemicals. Again, some of those are blood, but there is a mismatch between the CDC is able to monitor some of them through monumental effort, but what ones are missing, what are falling through the cracks. So at EPA, we being a government agency, we immediately developed an acronym. We have what we use is what we call just a systematic empirical evaluation of models. And so the idea there is very similar to what Nate Silver does with his election forecasting. There's lots and lots of polls out there. Some are good, some are bad, some are biased. If you carefully look at how each of those polls works, and you subtract off the bias and you weight the poll by how often it's right, you can come up with a consensus prediction. And another example that we have a lot of experience with is weather forecasting. If we had a single hurricane model that was really perfect, we would just run that hurricane model and weather forecasting would be very boring, but we do not have that. Instead, we have dozens of hurricane models, and parameterizations of even the same models. And so we kind of look at the range that we get. And so this is a storm back in 2017 that devastated the Caribbean. We knew that it was going to hit the Caribbean, and so you see a central tendency there. But then the uncertainty in the model predictions had it making landfall anywhere between Louisiana and Maine. That's the uncertainty in there. Now again, like I said, if we carefully characterize the uncertainty and we find that hazard, that all the people in North America live 10 orders of magnitude away, that uncertainty doesn't bother us, but we need to know that uncertainty. And so what we do is we take every model that we have that can make an exposure prediction, and we compare it with the biomonitoring data that we have that's available, and we hope that there's a correlation. When we first did this, there wasn't a correlation. So that drove model refinement. But then we estimate empirically what the uncertainty is, the scatter of the points. Never believe a modeler who shows you all their points along the line, because no model is perfect. And so one of the major limitations that we faced is really identifying, one last thing. Each point here is a different chemical. So DEHP might be a point here, bisphenol A is a point, various pesticides are points. We're evaluating across many, many different chemicals. One of the major impediments to this sort of research, though, is that we use DEHP very differently from how we use DDT, which we hopefully don't use, from how we use a pesticide. We need to know how every chemical is used. That's very tricky. You might think that this is not, but if I give you a database of 700,000 chemicals and ask you how they're all used, that's not a question we know how to answer. We don't have a giant list of how all these chemicals are used. Or a giant list might be 2,000 chemicals, which is a monumental achievement. It still doesn't cover every chemical that's out there. So what we have done is actually build machine learning tools that can take a chemical structure and do a best guess at what the probability that a chemical might get used. So we have models that can predict, this looks like a chemical that's used outside the home by industrial processes. This is a chemical that looks like it's used by pesticides. And obviously the focus here, we have a list of chemicals that looks like these might get used in diet. And that does include food contact chemicals, food additives, and then some manual curation of these chemicals are in food. And just in case you wonder how can we possibly do this, this is basically the same technology that Netflix uses to say, you know, if for some reason you like Nicolas Cage movies and you like action movies and you haven't seen The Rock, well, you're probably going to give it 3.5 stars. It's the same sort of reasoning. It's just done with large training sets. In the case of Netflix, it's millions of other people like you who've watched things. In this case, it's tens of thousands of chemicals that we know something about. This is all based on technology that actually, I'm sorry, the reference is down here at the bottom. All these slides will be public. Catherine Phillips, a colleague at EPA, published 2 years back, where you can basically take structure, and this shouldn't surprise any of the chemists in the room. We can take chemical structure and we can predict how a chemical is going to be used. Is it a viscosity controlling agent? Is it a hair dye? Is it a perfuming agent? We're doing the same thing here to predict, is it a dietary chemical? Is it an industrial chemical? So the ensemble of models that we're looking at, we got about a dozen models. We pulled together a team of modelers from around the world and basically tried to come up with the best predictions we could. So again, thinking of Nate Silver, taking all the polls and aggregating them, here are all our polls. And so some of these models model consumer scenarios, some of them this is just a list of chemicals that have been banned by the Stockholm Convention. And then we have pesticide models and we have dietary models. And we pulled all these together. And what we were able to find is that we can actually explain roughly 80% of the chemical-to-chemical variability in exposure for the NHANES chemicals. And so when we look across them, and some of these chemicals, sorry, this key is a little hard to read, some of these chemicals are just used in industrial processes, and then the chemicals that are at the high rate, so this is in mg per kg per day on a log scale, so some of the chemicals that we have high exposures to turn out to be in diet and in our homes that we have real familiarity with. So the model seems to work. So then we take those models, those dozen models, and we run them for every other chemical that EPA has in their database. We have a database right now, DSS Tox, has about 800,000 structures in there. We ran DSS Tox for all 800,000 structures. What we came up with was roughly 2,000 chemicals that had any probability whatsoever of having greater than 0.1 mg per kg per day exposure for the average population. So that includes a whole bunch of chemicals that the CDC hasn't looked for yet, but these are nominations for chemicals that we might want to study more. At the same time, we were able to show that we predict, and again, this is for the average person. If you're in a factory that makes this chemical, or you build your own swimming pool and fill it with one of these chemicals, I can't help you. The average person, though, we expect would get exposed at less than 1 microgram per kilogram body weight per day for 470-something thousand of these chemicals. So again, we've done a separation, a prioritization of which chemicals to worry about the most. Now we're here to food contact modeling things. So one issue that we had is as we looked across this, pesticides are really well covered. Industrial chemicals are really well covered. Modelers have been modeling these for a long time. We had USTOC's dietary scenario, which was adapted from a United Nations and CTAC model to dietary scenario. It turned out not to predict anything. And then we had one model that was predictive. So we have exactly one model that worked for food contact migration. And so this was a model published by my colleague, Kristen Isaacs led the research, and her former postdoc, Daria Burial, was the first author back in 2017. So what does this model do? Basically, it draws a line through the data that Dr. Hartman was describing earlier. So we took a data set provided by the EPA of migration of chemicals into food products, And we drew a line through there, and we were able to get an R-squared of about 0.7, so we're roughly predictive. And again, I'm comparing against in vitro studies. I just need to get a ballpark figure of, is this plausible, not plausible? When we look at how the model works, well, first off, at least my friend, hydrophobicity, or log P, is a big predictor. And then turns out the number one predictor of food migration was concentration in the material, which is good. That's a nice reality check. This is not to criticize this research. Basically, they're replicating 70% of the measurements by just considering concentration in log P. What we can then do is actually start to compare it with the NHANES data, and what we see is for chemicals that have either just consumer product, just food contact, or both exposure, we're looking at predictions on the y-axis here versus NHANES levels in blood. And we've got a pretty decent correlation. We're able to explain, again, for the average person, what the average exposure is using these two models. So how do we know about any of this stuff? Well, what we do know, the data sets we have are reported in a database called the Chemicals and Products Database. This is provided publicly by the EPA. It was described in a publication last year. And it contains all sorts of resources, material safety data sheets, consumer categories, the functional use data, all sorts of things. I'm going to draw a little bit of focus to some screening effort that we did on some measurement data. And this was analytical work done by Southwest Research Institute under contract from the EPA. So we know, and again, Dr. Hartman broke this down, you have targeted analysis, you know what chemical you're looking for, you have a radio-labeled standard, you look for that chemical, or you can do computer-enhanced analysis. We're trying to develop the tools to really understand computer-enhanced analysis. But what you get down to is, especially in liquid chromatography, you end up with a mass. That mass might uniquely match to a formula, if you're lucky, but that formula could match to lots and lots of structures. So how do we reason about that? One of the tools that we use for reasoning is, I think, relatively sane, is that we basically say, well, given the masses, given the formulas that all match that mass, how often is that chemical detected in other studies? What's the toxicity? So I want to rule out the really poisonous formula, or not formula, really poisonous structure before I rule out the others. What do my exposure models predict in terms of exposure? And so when we combine all that information, we're actually able to make a reasonable decision on what that mass might be. I should also say the EPA chemical dashboard is set up for mass spec right now. This is not the typical user, but if you want to go in there and you just have a mass, you can actually search a mass and a tolerance and get a list of every structure that we know of at the EPA that matches that mass. So we did a pilot study where we looked at 5 examples each of 20 different household items. And the goal of the study was not to be statistically representative. It's the opposite. It was to be methodologically challenging. So we're really throwing something at an analytical chemist and seeing what we could see with gas chromatography matching to the NIST, the publicly available NIST database. And so what you have here in nice Clemson colors are, I did not go to Clemson, I just noticed this, but you have the chemicals that were found in various things. So carpet had relatively few chemicals, whereas carpet padding had more, and cotton clothing had lots and lots of chemicals in it. We looked at breakfast cereal down here at the bottom. And then we have 2 colorings. One is purple. Here's the number of chemicals and hundreds of chemicals that were found by this screening analysis. And orange is the number of chemicals that we had in any database whatsoever. So of the 1,600 chemicals that we tentatively identified in this study, about 1,400 were novel. So that means on average, per product we looked at, we found 14 new to the EPA chemicals. If we look at food, so this was breakfast cereal, 5 different examples of breakfast cereal. We saw on average 90 chemicals in the food that we knew would be in the food, so that's including using the label on the packaging. And then we found a total of 120 that, again, we didn't have in any database. This isn't like we found bisphenol A. We know what bisphenol A is. We found bisphenol X, or something that we hadn't seen. The concentrations were typically, and I didn't know the 50 parts per billion rule, but typically down at 10 to the minus 2 micrograms per gram, so not so bad. All right, how do we believe any of that? I'm not an analytical chemist, although I'll do my best to field those questions. Well, like I said, Catherine Phillips developed a model that allows us to look at the structure and see what the heck, a chemical might be. And so when we do that, for instance, with baby soaps, we found a whole bunch of new chemicals in baby soaps. Well, that could seem a little disturbing, but as anybody who watched Breaking Bad knows, making something 100% pure is really challenging. So if you're making a surfactant and you've got something that's 95% pure, 5% of it's going to be something else. And what we typically saw when we looked at baby soap is that the other chemicals also look just like surfactants, just like perfuming agents. So what we're seeing is not that we're discovering weirdo chemicals and things. We're discovering similar related chemicals. But if you get the same secondary chemical in hundreds of products, well, then maybe we need to start looking at that and evaluating it. Maybe we're asking the wrong question. So just to wrap up, EPA is faced with dealing basically with everything that's not a pharmaceutical, not a food additive, and that's a lot of chemicals. And we really need new tools. And so people are often willing to throw bricks at in vitro methods because, oh, they're not as good as X. Well, we don't have even animal studies. I'd love a rat study for some of these chemicals. We do not have that for these chemicals. And so we are working on developing new approach methodologies to deal with these things. Now, I haven't talked a whole lot about gaps. I could do that for the next 6 hours if you wanted. But even though I'm explaining a lot of the NHANES data, that's really only 114 chemicals. That's a lot. That's a lot more than 2. But if we're extrapolating to 800,000 chemicals, I'd feel a whole lot better if it was more chemicals. We also have a lack of dietary exposure models. Now, these models need to be run for 800,000 chemicals, so it's not like you can spend all day parameterizing it for your favorite chemical, but we do need models like that. And then we actually, as we train our machine learning models, just like with Netflix, it's really important to know what movies you like, but also what movies did you hate? It's really hard to train a pathway model to predict things that are in food if we don't have examples of things that aren't in food. But you don't find a lot of research papers of this is my list of chemicals that I did not find in food. But that's exactly the sorts of information that we need right now. So I just want to, like I said, this is dozens of researchers within the Office of Research and Development. Kristen Isaacs, my co-lead, was the lead in developing the migration model. And then we have lots of collaborators outside here. All these slides are publicly available. So just ask me for them or they'll be distributed. And then anyone with an asterisk here, a little hard to read against the background, is a trainee. We work with lots of trainees. I'm very happy to work with trainees as colleagues when they move over to this list over here. So they're all very good. And if you're looking for somebody, let me know. References are also provided and I will not read them. Thanks.
Thank you very much, Dr. John Wambaugh, EPA, National Center for Computational Toxicology.